Panel design is a true art form in flow cytometry. While this video will not make you an artist, it will give you the tools to start your journey. Welcome back to the Intro to Flow Cytometry series. If you're joining us partway through the series, you can find the link to the rest of the videos in the description. In today's episode, we'll be talking about panel design and how to properly select fluorochromes when designing your flow cytometry panels. Now, does your panel design kind of look like this? A random hodgepodge assortment of markers with fluorochromes? If so, we can probably improve your resolution. Now, before we go too deep into panel design, there is one important thing that you must always include in every panel: a viability dye. No matter what you're working with, no matter what cells, no matter what you're staining for, always make sure you have a viability dye in any panel. Dead cells will destroy your data, so you want to make sure you've removed them. The nice thing about viability dyes these days is they can be put in pretty much any channel. I recommend putting them in channels that are generally underutilized. This may be, for example, the UV channel, where generally there are not a lot of fluorochrome selections available, or say the far reds, where you may not have the best resolution of your markers. There are also other channels that are generally not that great for fluorochromes, like the AM-cyan channel. So look at those channels first when selecting where to put your viability dyes. When we think about panel design, we can really break it down into 6 general levels. Less than 6 fluorochromes, between 6 to 10 fluorochromes, or more than 10 fluorochromes. Kind of like beginner, intermediate, and advanced. Now, these general breakdowns are for the instruments that I have in my facility, where we really can't run more than 18 colors. If you're working in a facility that has an instrument with higher fluorochrome options, your breakdown might be slightly different. However, the theory behind panel design will remain the same no matter where you make your breakpoints. When you're dealing with designing a panel for 6 or fewer fluorochromes, the easiest thing to do is pick bright fluorochromes for every channel. So what do I mean when I say a bright fluorochrome? This really is just related to the stain index of the fluorochrome, or the amount of space that you have between the negative and the positive. The wider that gap between the negative and the positive, the higher your stain index will be, therefore the brighter the fluorochrome is. So essentially, the brighter the fluorochrome, the better you'll be able to resolve that signal. So when you're dealing with fewer than 6 fluorochromes, there are more than 6 bright fluorochromes on the market. So you might as well select the best fluorochromes possible and not use the ones that have a lower resolution capacity. So the graph to the side here will give you some idea of how stain index looks. You can see that as we change fluorochromes, basically our gap between the positive and negative is becoming smaller and smaller. So the brightest fluorochromes that have this greatest separation are going to be things like BV421 or PE. Down at the bottom of the list, you'll see the fluorochromes that have worse resolution. After you've factored in fluorochrome brightness, the next thing you want to do is reduce the spillover. Essentially, this means spreading your fluorochromes out as much as possible. The easiest way to do this is starting by selecting a fluorochrome off of each laser line. Generally, the first fluorochrome off of each laser line will be the brightest one. So if we have a standard, say, 4 laser instrument, we'd want to choose the first fluorochrome available off each of the 4 lasers. And that would give us a really easy way to build a 4-color panel. That being said, we can still add more colors to this panel. So even though we might have multiple fluorochromes on a laser line, we can still reduce spillover by spreading out them across various filters. For example, if we look at our 561 nanometer laser line here, you can see we have a number of various filters coming off of that laser line. So we know PE is a bright fluorochrome, so let's select that as one of our fluorochromes of interest. Now, moving down this laser line, there are another few bright options, most notably PE Sci 5 or PE Sci 7. So if we want to spread out our fluorochromes the furthest, which would make more sense? If you selected PE Sci 7, that's great, because PE Sci 7 is further away from PE, so there will be less overlap between the emission spectra of these 2 fluorochromes, and therefore less compensation. So these are the things you can start thinking about when selecting your fluorochromes when you have 6 or fewer. So once you've selected your 6 fluorochromes and got them into your lab, the last and final step of any panel design is to test your panel. Now, what do I mean by testing a panel? Well, essentially, you're going to stain your cells as single stains for each of your colors, and then do one that's all of your antibodies together. And what you're looking for in each of these samples is if the biology makes sense. So for example, if you expect that one of your markers will be about 20% of your population, but when you stain your samples you see it's 80% or 5%, this doesn't really make sense. In that case, you'd want to go back and re-troubleshoot or potentially even rebuild your panel. Now, other things you can look for when testing your panel is how good the resolution or the separation looks. So if you notice on one of your markers you have much poorer resolution than you had expected, as in the positive is much closer to the negative than you would like, this may be something that you want to replace. And so you could go back and look at different antibodies that may be available for that antigen. Other things to keep an eye out for are diagonal populations. So when you plot 2 fluorochromes against each other, if you have a fluorescent signal that basically travels all the way up the plot at a 45-degree angle, that's generally not a good thing. Very few markers are that closely co-expressed that you will have a fully diagonal population. So you'd want to look at your panel design and look into that a little bit further. And finally, in your sample that's stained with all of your fluorochrome combinations, you want to keep an eye out for biologically questionable or biologically impossible populations. What I mean by this are 2 markers that really should never be co-expressed. So say you had a marker for T cells and a marker for B cells in your panel. Now if you found this marker co-expressed on a single cell type, that's not something that really you would expect to happen. The T cell marker should be on T cells, the B cell marker should be on B cells, and they should never be together on the same cell. So if you see something like that in your panel, that's a good indication that you need to go back and redesign. So now that we've tackled the beginner stage of panel design, as in less than 6 fluorochromes, let's move up a step to a more intermediate panel design and look at what you need to do when choosing, say, 6 to 10 different fluorochromes. So this starts with the same basic logic. We want to start with bright fluorochromes. However, in this case, we want to make sure that these bright fluorochromes are put on the antigens or on the markers that are going to be the hardest to detect. So these are going to be things like markers that have a low level of expression, markers that are on very rare populations, markers that are extremely important to your assay, so things like cytokines that are generally harder to find. You want to make sure that these are given the brightest possible fluorochrome option so you have the best possibility of resolving them. So this is when you can get into classifying your antigens into groups like your low expressors or your very important expressors versus your high expressed antigens, things like parental markers, things that are on and off, the antigens that are really easy for you to detect. And once you've classified all your antigens into these 2 groups, that's when you can start assigning fluorochromes. So for your more difficult low expressed antigens, you want to give these ones always the brightest fluorochromes. And then for the rest of your antigens, you can start moving on to the dimmer fluorochrome options. Now, one very important thing to note is if you have any intracellular markers in your panel, no matter what your panel design or the number of fluorochromes you have, you always want to make sure that you give these ones not only a bright fluorochrome, but never give them a tandem fluorochrome. Fluorochromes are already very large in nature. It's like attaching a giant beach ball to an otherwise small antibody. So if you're using a tandem dye, this is like sticking 2 beach balls on that antibody. And it becomes infinitely harder not only to get into a cell, but also to get back out of the cell if it hasn't bound something. So when working intracellularly, always avoid tandem dyes. If you remember from our last section, when we have 6 or fewer fluorochromes in a panel, we want to try prevent spillover. So this is talking about compensation. Now this also applies when we have 6 to 10 fluorochromes, but we need to now factor not only spillover, but also spillover spread. Now, we do have another video dedicated completely to talking about compensation and spillover spreading error, so make sure you check that out for more details. So spillover spreading, or spreading error, is a spreading of the background. Now, this can be problematic because it will effectively decrease the resolution. So as you can see in the plot here, because of the spread of our PE positive population, we would have a very difficult time detecting a dim PE Psi5 signal because this would fall underneath that spread population. So again, this spreading error would then result in effectively a loss of resolution, so an inability to really discriminate your populations. This may sound very dire, but it's actually fairly easy to figure out if you're going to have spreading error. And this is simply done by making a spreading error matrix, or spillover spread matrix, or resolution impact matrix. It has many different names depending on the software you use to calculate it. Generally, because we're using FlowJo, we call it a spillover spread matrix. Now, to make a spillover spread matrix, all you need are your standard compensation controls. And again, we cover this in detail in the other video, so I'm not going to go into it here. That being said, there are resources available where if you haven't made your own, you can look at a stock resolution matrix or spillover spread matrix to determine if you might have spreading error when making your panel. So to the side here, you'll see what a sort of stock resolution impact matrix would look like. So this comes from a company. Again, this is not done with your Fluorochrome, so may not exactly reproduce the spreading error you'll find, but will give you an indication of where you may find spreading error. Now, you'd want to avoid combinations where you see red. That's going to be your worst spreading error. And depending on how many fluorochromes you need to have, how many options you have available on your machine, you might want to stick in more the green-yellow side and completely even avoid the pinks. So if we knew that our APC was going to be a low-expressing antigen, which fluorochromes should we not add to this panel? So if you selected BV650, that is correct. Now, if this was your panel, you may also choose to avoid things like PerCP Psi5.5, BV711, or Alexa 700, as these also may have some impact on your resolution. Otherwise, any of the other fluorochromes present would be potentially a good combination for your panel. And finally, just as in the less than 6 fluorochrome panel design, the last step is to test. And again, look for those biologically questionable, biologically impossible, or just weird responses that you might see. Your panel design will almost never be perfect on the first go. Generally, you will need to go back and revamp your panel. So just keep that in mind, that your initial panel design is just that, the initial design. There will be iterations necessary until you have a great panel to work with. After the 6 to 10 color intermediate panels, we start to creep into the advanced panel design area. This is the more than 10 fluorochromes. Because this is an introduction to Flow Cytometry series and almost nobody starts with more than 10 fluorochromes in their first panel design, we're not going to go into this here. So stay tuned for a future advanced panel design workshop where we'll look at various tips and tricks for dealing with these high parameter panels. To give a quick recap, when designing your panels, always start with the bright fluorochromes. If you don't have very many markers, you might as well stick every marker on a bright fluorochrome. If you have more markers than you have bright fluorochromes, start by putting the most difficult markers to detect on these bright fluorochromes so you have the best chance of seeing them. And keep in mind, if you have any intracellular markers, do not put tandem dyes on these markers. Next up, you want to factor in spillover and spillover spreading error. So try to spread out your fluorochromes as much as possible while looking out for areas where spreading error may happen on your instrument. And finally, but very much most importantly, always test your panel. A panel may look perfect on paper, but in practice, there may be issues still on an instrument, and you will not know this until you test your panel. So before you start diving into your huge experiment with your irreplaceable samples, test your panel to make sure it's going to work. And with that, thanks for joining us, and we'll see you in the next one.